Epithelial cells form the barrier between the outside world and the interior of the body. This barrier is critical for defense against microbes like bacteria and viruses, as well as for communication between the tissue and the outside environment. For people with cystic fibrosis, the disrupted function of CFTR in epithelial cells can lead to disease in several parts of the body, especially in the respiratory system, where chronic bacterial infection and hyperinflammation lead to decreased lung function. Hi, I'm Travis Ruch. I'm a postdoctoral fellow in Joanne Engel's lab at the UCSF School of Medicine in San Francisco, California. Through the support of CFRI, I'm currently investigating how the bacterium Pseudomonas aeruginosa interacts with epithelial cells. Pseudomonas aeruginosa is the most common damaging lung infection in CF patients. The presence of Pseudomonas in the lung is associated with increased inflammation, decreased lung function, increased hospitalizations, and worse clinical outcomes. In the CF lung, Pseudomonas can grow as a community of bacteria known as a biofilm. Forming a biofilm likely grants Pseudomonas a growth advantage. Also, growing as a biofilm provides the bacteria with increased resistance to antibiotics. Biofilm growth occurs on top of epithelial cells, so learning about how Pseudomonas and epithelial cells behave in this context is of great interest. To understand how Pseudomonas interacts with epithelial cells, it's important to know the basic architecture of this cell type. Epithelial cells have a segregated or polarized structure, with the outside-facing top of the cell being distinct from the interior-facing bottom of the cell. We refer to the top of the cell as the apical membrane and the bottom of the cell as the basolateral membrane. Keeping these parts of the cell separate allows them to take up nutrients from below and secrete mucus and monitor for microbes from above. It is not well understood how or if epithelial cells respond to early biofilm formation. Work in the Engel lab continues to investigate this gap in knowledge. We employ a cell culture model to study the interaction of Pseudomonas with epithelial cells. Using this simplified system, where only Pseudomonas and epithelial cells are present, allows us to closely monitor their interaction. We work with both normal and cystic fibrosis epithelial cell lines. This comparative model allows us to examine how the loss of CFTR function impacts the response of epithelial cells to infection. In order to mimic the physiology of lung epithelial cells, our lab-grown cells must be polarized. However, most cells grown in the lab are not polarized and simply spread out onto a plate. To avoid this, we grow our cells on porous filter supports. This allows them to take up nutrients from the bottom and receive the necessary cues to polarize. Once the cells are polarized, we infect them with Pseudomonas. The strains of Pseudomonas that we use in the lab make a fluorescent protein so that we can observe them under the microscope. Following infection, we prepare our samples for microscopy analysis. We employ state-of-the-art imaging techniques to examine our samples. Epithelial cells are very tall and therefore require a three-dimensional scanning process to image. We scan through the samples with lasers to highlight specific parts of the cell. Once scanned, we use computers to assemble the scans into three-dimensional images.
Exactly.
This process is similar to a CT scan, but at a much higher magnification. This technique allows us to monitor changes in the cell caused by Pseudomonas with very high resolution. We have found that in response to early Pseudomonas biofilm development, polarized epithelial cells form a finger-like projection extending out of their upper apical membrane. Surprisingly, this structure has characteristics of the bottom basolateral membrane, suggesting that the cell has inverted its polarity underneath the newly developed biofilm. We have termed this structure a protrusion. This response occurs rapidly, as can be seen by the green flash underneath the red bacterial aggregate in this live cell time-lapse movie. Strikingly, the changes in cell polarity are associated with activation of the immune response. What this means is that the binding of bacteria to epithelial cells causes the top of the cell to pick up characteristics of the bottom of the cell, and that this change kicks off an alarm signal that warns of an incoming bacterial infection. We hope to determine how the protrusion pathway functions in the context of cystic fibrosis cells, and if it contributes to chronic colonization by Pseudomonas. We are also trying to identify specific bacterial and human proteins involved in this process. Understanding the protrusion pathway at a molecular level may reveal novel drug targets to treat CF disease and improve the quality of life of CF patients.